for the analysis we use about total 315 population, the training population, about 5,000 F5 plants, genotype using LD dart, markers, because when we genotype, there are two different methods when we use genotype, because when we're talking about the 5,000 plants, I cannot genotype them with the high density or those high cost genotyping systems. So then we start to talk with the dart. They come up with a new technology called LD dart, which is low density dart. But we got challenged because markers were different. So we started to find marker based on sequence similarity. So we find about 900 markers which are common. And then we start to use merge by sequence and start to those things. If you see this image, this shows the similarity of those 5,000 lines with the training population. This is the training population screen, and these are the different lines among those things. So these lines, some of those lines are closely related to these training populations, some are not closely related. So this give us a lot of understanding of how GS should work in the regular breeding program. And when we were doing the prediction accuracy, we got very good prediction accuracy for yield rate like 100 seed weight. But seed yield also was a good prediction of about 2.2. Many people say, oh, 0.2 was very less. But when it talks about genomic selection, 0.2 is not considered bad. It's an optimum, and we got good prediction accuracy, although there are some challenges. So we started to work with Dr. Bhardwaj and then AICRP system in India to address some of those challenges. And now we are together with them are deploying these breeding program in the regular breeding program, this genomic selection. Apart from this, then we started to work on the how this relationship metrics work. So we had some of the data we were having. We started to use for deployment how this different relationship metrics work. So for this, what we did, we used this training population 320 as a cross-correlation of the training population, ICRISAT IRE material. And then for the testing set, we used those 5,000 1 and 2 genotypes from the ICRISAT and magic population from ICRISAT. We use this as a testing set, and we started to understand the influence of marker density, population structure, and the relationship on the applicability of GS. So this was a separate experiment to test those hypotheses. If you see this PC results, it summarizes that PC component 1 and 2 explains almost 80% of the variance among those populations. And this showed that 100 should contribute at least by variance component. We started to analyze those things. So this is the heat map. This shows that we identify a common set of markers between training population as well as testing population. Those 5,000 markers as well as 320 lines and the magic population. We identify about 28,000 common markers, which are common between training population and the testing population. These are distribution on the genome. We test those cross-correlation scheme, as I mentioned, within site CV1, across site CV2. As I mentioned in the previous slide, we use 6 different models: E plus L, main effect to environment and line; E plus G, main effect to environment and marker information; E plus L plus G, main effect to all three major factors: environment, line, and marker information. Then we use the interaction of environment into line interaction, environment to marker interaction, environment to line into environment to marker interaction. All that together we use. And we got these kind of prediction accuracy. If you see, when we are including the line, environment, and all the interaction, we were getting very good prediction compared to only line and environment. And this, we started to check for this. And if you see, average prediction accuracy were different for DTF and unseeded, and for pH and CY, especially for the models compared to GWAS. So this is very good that GWAS inclusion models are very good. Complex trait like seed yield, it has lower prediction accuracies compared to these two flowering and plant height, just simple traits. And here, if you see the prediction for different traits, and they were very showing the optimism that yes, we can test those things. And when we start to check those things, if you see the models based on the interaction of EL and E by G were given the very optimum prediction accuracies, and we started to use these models for the further testing of our genomic selection model. This is genetic diversity based on the training population profile, and if you see, more than 6,000 genotypes were used for NG tree, including 5,000 FI lines, about 1,200 magic lines, and 300 training population. This clear cut shows that there is a training population is blue in indicators here, and these are the FI progeny which are mainly desi, and these are the magic lines. They may clear cut difference in the based on these markers. These markers are correct, and they were able to segregate those things. Then we started to use it deployment strategy. So what we did, we selected those 320 as a training set. We used two sets of marker data. One was 28,000 common SNPs. Another set was 15,000 selected based on the GWAS. We did GWAS from these markers and selected markers which were found associated. Test set was used, about the 6,000 lines were used as a test set. And then predict F5 Lausanne for the prediction use and prediction of magic. We made separate predictions. In the first strategy, we use all the common markers, that is the 28,000. And second strategy, we use subset, about 15,000 markers based on the GWAS. And we compare the result of two strategies to train the model. And if you see, we got these mean prediction accuracies, which were for the days flowering range from 0.4 to 0.5. And for plant height, it ranged from the 0.3 to 0.38. Similarly, for seed yield was in the range of 0.2. So based on these comparisons, we find that initial results from price says that GS has definitely a role in accelerating the chickpea improvement effort. For prediction accuracy, like seed yield was lower compared to the days to plant height. As I mentioned previously, there are some challenges about the relationship. So we started work with Dr. Bhardwaj and All India Chickpea program. We added some additional 500 lines from national breeding programs as training population. This year, the training population evaluated at different locations for yield and quality traits, and we are expanding the diversity by including to increase the genetic gains using genomic selection. So this is about genomic selection, and this comes the last point of the decision support tools as I mentioned. I mentioned briefly about the GOBii. This is Genomic Open Breeding Informatic Initiative. This is a global community and a Gates-funded project which was led by Cornell University and 3 CG centers, ICRISAT together with IRRI and CIMIT. GOBii focused on 5 crops: chickpea and sorghum from ICRISAT, rice from IRRI, and wheat and maize from CIMIT. Apart from this, some of the partner centers are DART, James Hutton Institute UK, and BTI in the Cornell. So mission of this thing was to develop a stable, high quality, easy to deploy genomic data management and develop these decision support tools and release to the breeders and improve data availability so that breeding management system can make this. GOBii, working together with different partners, developed 4 tools: GDM, GOBii Genomic Data Management, GOBii DART tools for making selection, quality control selection, GOBii Genomic Selection Pipeline, GS Galaxy, that is used for the genomic selection these days, GOBii GHI tools for the MABC module for Marker Assisted Backcrossing, and F1 pedigree verification module for making the hybrid testing in those things. Just a brief about the Galaxy pipeline. GOBii together with EIB developed customized the existing GS pipeline where you have all the models that you can calculate. Blues calculator, encode B2 genotypes, existing. You can impute your genotyping data and you can do the GBS prediction, you can do cross-validation. For this, what you have to do, you just upload a genotyping file, you upload a map file, you upload a phenotype file. If you don't have map file, it's fine. For genomic selection, mainly you need genotype and phenotype file. Once you upload those things data, you just execute. And what is the steps? If you see, GOBii data extract post to Galaxy, and then get data upload from this to browser to the server, and then it runs VGL calculator for calculating genomic predictions, and then visualize the tool, and you can save the workflow and go back later. In addition, GOBii also provide integration with the different systems like BMS at ICRISAT for the breeding management system, GOBii, as I mentioned initially, for the genomic support. So GOBii has integrated itself with the GOBii, BMS, STPG, so that marker data come directly from the vendor, come directly to the GOBii, and using those support tools and using BMS phenotyping data, the breeder is able to make decisions. So overall idea was that contribute GOBii is to address the concerns, provide solution to the breeder. They should not be handling the Excel files. And from ICRISAT, major focus was chickpea and sorghum, but now for new crops, other mandated crops like groundnut, pigeon pea, palmet are also being added. So these are some of the resources that ICRISAT is putting for the GOBii. In the end, agenda was that translating genomics information crop. So a lot of genomic resources and costly genomic platforms are made available with precise genotyping, and then user-friendly pipelines and support tools developed, and these are being deployed using different tools, mass genomic selection for the increase in the breeding program. In the end, I would like to suggest that now, instead of this earlier thing, this is one of the came from this year review we made it, that now it's time that we should start talking about the sequencing-based holistic breeding approach instead of the regular breeding program. So what we need to do that we need to start using those omics, genetic resource populations, and like GWAS, identify the superior alleles based on the sequencing, phenotyping data, and those things. And then start using them in the breeding population based on sequencing, phenotyping. This information helps in the parent selection. We make the F1, we do the early scanning, and once we reach this F6, we select lines based on high GBVs, and we start the yield trials and develop the improved lines. Now, future will be a integrated toolbox, not the isolated one. If we have to make a good program which focus on the BMS, GOBI, genomic resources, and genetic resources, and then these are used together with agronomic practices to develop the improved varieties. I would like to end with the quote from Norman Borlaug: "Food is the moral right of all who are born into the world." But that in that scenario, food was considered as food. But to this, I would add that now we should not talk about only food. We should be talking about the food with nutrition. So we should be working provide to the food. Another thing people say that everything can wait, but not food. So we need to give due credit to the food. And thanks to all the colleagues at CG, special thanks to Dr. Rajiv Varshney for giving the opportunity to work here and contribute to Chickpea, and donors, Bill and Melinda Gates Foundation, Government of India, ICR, GOBI, and ICRISAT. Thank you. If you see, there are not many reports available. These are initial insights and some of the things which I also came to know recently. But right now, we need to assess the very first available diversity what available in Chickpea to increase the first. There is a need to have the focused efforts. What kind of diversity for beta carotene available in Chickpea? In general, it was higher to golden rice 1, but yes, current status, general status is not higher than golden rice 2, which increased 23-fold of the golden rice 1. So this is in preliminary stage. We are just checking, and once we are developed gen diversity, and it's not a kind of competition that we have to beat golden rice. No, we have to see first available diversity and then see what scope we can do. Second, is it effective to how to use low density sequencing genetic gene fitting programs? Is it enough to make the marker the whole genome? About the low density sequencing, yes, but there is a clear strategy you have to follow. You cannot straightaway use, because I skipped one of the slides in event of time. When to use the low density? Suppose I need to use low density in a population. So general strategy, what people are doing these days, that all the parental lines has to be sequenced on high coverage, 15 to 20x minimum. And if you have high coverage data available for those parental lines, then progeny is using low coverage, you can predict using imputation and different algorithms. So those are possible loaded. But in addition, if you're talking about the genomic selection using 5,000 kind of marker, so earlier people used to talk about, oh, for genome selection, I need millions of marker or 80,000, 100,000 markers. But based on our experiment, based on different groups who are working on those things, they suggest that there is no need of these many markers. If I say Chickpea, I don't need more than 2,000 markers to run the Chickpea genomic selection algorithm. So that's the reason we thought to develop about 5,000. In any population, we'll get about 2,000 to 3,000 polymorphic markers, and they will be ideal for us to run a genomic selection program using those 5,000 markers. Question 3, why genomic selection is the most promising for genetic improvement? It's not that it's considered most promising, but as I showed you, if you see the breeding equation, there are 4 parameters. Genomic selection in one go, if you do it properly, you get optimize your prediction model. So they address several of those factors, because it's always expensive to take few thousand lines to the field. You cannot think about taking 5,000 lines or 10,000 lines of field, because that adds a lot of cost. In national system, we don't care about the field cost. In CG center or other center, if you see the private industries, they calculate each and every cost, labor cost, field cost, and everything. Even in the national system, you don't pay for the field charge, but a lot of management goes. Scanning 10,000 line is always challenging. Recording of data, when 10,000 lines are in the field, you cannot estimate 10,000 line data days to even forget simple days to flowering. It will not be easy for, this will be a lot of work and there's possibility of error. But if you can do that thing based on marker, this will definitely help. And so that's why gene selection has the potential for improving. But there is one caveat. To deploy high quality genomic selection or most effective, first thing is you have to have the precise phenotyping data. If you don't have phenotyping data, good phenotyping data, you cannot have a good genomic selection model. So this is correlated thing. So, and nowhere I'm suggesting that when we have genomic selection, we don't need to do phenotyping. We have to do phenotyping, and phenotyping is key for running a genomic selection program. How can we assemble various scaffolds of sequences? How can we remove repeated SNPs in each scaffold? So this will be technical. If you want me to address, or I can address this thing in the email. How can we make treatment with this? In the short period, can we make trait mapping with the comprehensive samples? For this, if you see 2019, there's a paper from Dr. Rajiv Varshney et al., the sequencing-based trait mapping. Now there's an approach, you can start using the bulk segregate analysis. You don't need to do the phenotyping of those things. If you have the phenotyping data, you just can select the extreme bulks for traits. Suppose you have disease resistance mapping population, you can select the trait which are completely resistant, lines, 10 lines from the resistant, 10 lines from susceptible. You can bulk select, sequence those two bulks to the parents, and using those BSA approach, you can do the trait mapping in a very short period. You don't need to genotype entire population. You don't need to do phenotyping also. If you have to improve the yield, what is the correlation between protein content and yield? The content of chickpea. The correlation between protein content and yield, there are reports that they are negatively correlated, and that's where genomics comes in picture. If we can identify that linkages, and genomics can, because when we are talking the high resolution mapping, high resolution, because genomics gives you information on the each base. This will definitely help to break that linkage, and we will able to break that thing. But thing is, this is theoretical. We need to work on this, and we started to work in this direction. But right now, I'll not say we have done it. This is something ideas need to do, and we are working on this direction. What was the age of chickpea plant used for DNA extracted for genotyping? Generally, we take 2 weeks fresh leaves, but when it is early, we can do just seedlings from the even 1 week plant, depending on the yes on without land races. Genomic selection is not for the land races. Genomic selection, if you see genomic selection, there are two strategies. One is the population improvement, one is the product development. For population improvement, where you want to improve the parent, many times you want to bring some of the land races to bring apart. But for the product development thing, genomic selection is mainly focused on developing the breeding population, where you have the parental line. So those things doesn't work on the land races. Too time consuming compared to the hypotype second half of the hypotype sequence. SNP is not time consuming. In fact, I'll say it's time saving, because if you do sequencing, you have to do a lot of sequencing analysis. You have to clean the data, you have to do those things. But SNP, once the data is there, you have to put this in the tool and you'll get the allele matrix. So this is not time consuming. The target plant has lost any essential characters. We want to reprogram this technology is less costly. There are different biometrics tools. So for different things, and depending on the sequencing data, there are different tools. For GWAS data, we normally use command line. We use GAPIT or the TESL, but there's command line. But depend on the data. So it will be difficult to recommend which tool right now and which tool not. It will be challenging, because this will be a lot of depend on the data quality, data coverage, and all those. How can you say which type of markers to be used? As one of the slides when I was talking about the markers depend on the application. First, many people labs are still using SRs instead of IM, although I said that we use SNP marker, but many people, because these needs a lot of capital. But right now, these are days you don't need to create infrastructure in your lab. You can easily outsource those things. Some of those technologies, in fact, at ICRISAT, we have CGSB, which is headed by Ms. Anu Chitaknani here. If you need some of the genotyping services, you can contact her. You can write an email to her, and she will be able to provide. So markers depend on the application and cost. How much cost you want to do was, that's all from my side. If I missed something, so you can tell me, Neha.